And we are now recording. So hello, everyone. If you have any questions, we would love to take them. Do you have anything to say, Sam?
No, I don't think so.
I'm very pleased with how many people are here. Oops. Oh yeah, Joseph wanted the weekly quiz and they were the first to ask, so we can go for it. Yes, okay. Quiz number 9. So what did we think on, okay, number 1, in the secondary structure of DNA, someone had a question. And here in the, oh yeah, no problem. And it's also being recorded, so don't feel like you're terribly missing out on anything. I missed the other review session. Is there? Go over the other quizzes? Rami? Oh, yes. And, I am not the guardian of the secret quiz questions. If you would like to rely on the generosity of the people you're in class with, I'm sure that they are generous enough to share it with you. I am not the secret question police. I had different questions on the quiz. I think it's a pool. They're probably just presented in a different order, Joseph. So we will go through them one at a time. Or I guess obviously you would know if you had this question or not. So yeah, I guess it is a pool, but I can try and get different questions too, I guess, by retaking the quiz. Anyway, so in the secondary structure of DNA, let's identify. Did we even talk about this? Anyway, so DNA has several different degrees of structure. And we learned about this in Bio 213. So, like proteins have first degree structure, then second degree structure, third degree, and fourth degree. The first degree structure of a protein is the amino acid monomer units that it's made of, the individual building blocks. And DNA is kind of the same, except the individual building blocks are the individual nucleotide units. Yes, that's the first degree structure. And then the second degree structure, yes, Casey, that's just for proteins in general. For DNA, the primary or first degree structure is the nucleotide base pairs, because those are kind of like the monomer units that hold the information that DNA contains. But then when you zoom out with an extra degree of complexity, so actually I should maybe even write this, and I don't know why I keep doing that. No, please, stop. Okay, so we have our first degree structure. That was not good. We have our first degree structure, and that is the individual units that something is made of, the A's, T's, G's, C's, C's, and A's. And then when we get to our secondary structure, it is the combination of all these, how they fit together. And that's when we start to see the information appearing. Because we have our base pair and its complement, it's complementary base pairs. So in the secondary structure of DNA, now that we know what it is, hydrogen bonds occur. Which do we think is the correct answer? D, the last one, D. I would agree. Hydrogen bonds occur between complementary, to connect bases on complementary strand. I should read the answers before I verify yours. Definitely not the first one, because we know that we have our base pairs, pretend that's a straight line. So like we have an A here, which means we'll have a T here, C here, which means G here, G here, which means C here. And we know that hydrogen bonding is what really connects these nucleotide bases together. And I can draw out the like 3D structure of the purines and pyrimidines if you want, but this doesn't happen anywhere. We don't have this G hydrogen bonding to this G. That doesn't happen. So it does not happen within the same strand because this is a single strand. G does not hydrogen bond to G. So we know that this cannot be correct. To connect sugar and phosphate groups of different nucleotides. So we remember also that we have our basic structure of DNA, where we have our 1, 2, 3, 4, and 5 prime carbon. This 5 prime attaches to a phosphate backbone. This 3 prime carbon attaches to a phosphate backbone. Oops. Backbone. So, and then to this first prime carbon is attached the nucleotide base. A or G, T or C. So to connect sugar and phosphate groups of different nucleotides, that doesn't even make any sense. We don't need to read that. Connecting the sugar or phosphate groups to different nucleotides, no, the sugar and phosphate connect to each other as well as to the nucleotides. There's no hydrogen bonding going on there. To connect bases on the same strand, we already verified earlier that that does not happen. So yes, like you all said, the answer is, can I get the pen away? D. So in an RNA molecule, hydrogen bonds occur to connect the nucleotides on the RNA strand. True or false? What do we think? Is this the right quiz? Oh no. Wait, should I be doing the quiz 8? Have you guys, oh no, there isn't a quiz this week, so we're good. Okay. False, false, false is what we see. Okay, thank you. And another false. So let's talk about it. Let's think about it. Sorry, the chat's going pretty fast. There's only phosphodiester linkages. It's the correct quiz. Okay, thank you. I say true. Kinga says true. So we have some trues and we have some falses. So true because of bonding with itself. I had different questions. So in an RNA molecule, hydrogen bonds occur to connect the nucleotides on the RNA strand. I love what Maya said earlier. So we know what is one of the big differences that we learned in Bio 213 between DNA and RNA molecules. There are a few big ones. Yep, Carla said it. And Casey also said a very important thing. RNA is typically single-stranded, whereas DNA is usually double-stranded in a helix. So RNA will have base pairs that are not usually connected to a complement on the other strand, like DNA would, you know? Also, Casey made a very good point that in the structure, we have our DNA and RNA. Obviously, we have the 1, 2, 3, 4, and 5 prime carbons, same on here. The thing is, on RNA, on this 2 prime carbon, we have a hydroxyl. And this pentagonal sugar is called ribose, and that's why it is called ribonucleic acid. Deoxyribonucleic acid, on the other hand, does not have an OH. It has been deoxied, and that's why it is called deoxyribonucleic acid. So Casey made a good point. That is important. But I think the thing that this question relies on is the fact that RNA is usually single-stranded. Because, RNA is going to be, it's going to make up your ribosomes, and it is going to be used in a lot of your most important cellular processes. It's the short-term molecule that we use to directly build our proteins and our organelles. DNA, on the other hand, is the long-term information storage. Someone in lecture made a really good metaphor that I can't remember, but RNA is typically single-stranded. We don't want to waste twice as much material building a double-stranded RNA if it's going to be the short-term molecule. So hydrogen bonds are not made because there is no complementary strand for the nucleotides on an RNA to bond to on the complementary strand. So I would say that it is false. Can you repeat what it said about me? Yeah, and Joseph has his hand raised. Then I will answer Casey's question. What's up, Joseph?
Yeah, so the reason I put true on this one is because we learned about something called secondary structure.
Okay, well, while Joseph is typing his answer or question, I will answer Casey's. So the durability of genetic molecules. Oh, he was talking? Can I not hear anyone? Have people been talking this whole time? Oh my God, that's so embarrassing. I had my volume all the way down. Okay, so please forgive me if you said a single word and I was not answering you. I could not hear you. Okay, so hi, Joseph.
Okay, so yeah, the reason I said true here was because we learned about something in class where RNA can fold on itself. Actually, Dr. Chiari made a big deal about this and how it can bond to itself, right? So the wording on this question can be answered in that way because the question is really vague. It says connect the nucleotides. Because if you think about it, right, when you have the hairpin structure, two bases or two nucleotides will connect to each other with hydrogen bonding.
Yes, you're right, because RNA does fold on itself like this. This is an example of tRNA, transfer RNA, that we use if we can remember the central dogma of biology, that DNA is translated into RNA, or DNA is transcribed into RNA, which is translated into protein amino acid monomer units. And during this phase, when we are translating, RNA, a special kind, transfer RNA, folds up onto itself. And all these lines I'm drawing are individual nucleotides. And so yes, they do connect to each other, but they are not bound together. This is a spliceosome, and it is also made of what is called SNRPs. I'm sure you all remember. Single nucleotide, I don't remember, but this is all RNA as well. However, it's just kind of folded and bent on itself. There's no, oh wait, did I misread the question? Yeah, there's no bonding between these because hydrogen bonding is a very strong chemical intermolecular interaction. And the reason that we have hydrogen bonding between these is to keep these two strands held together. But RNA is our our short-term molecule that we need to be really easily read. So would we want some really strong intermolecular forces between the nucleotides in RNA if we just want to get at the information inside really quickly to build our proteins? Yes, Casey, it's called the hairpin structure is what I drew. Very good.
I think I will still stay with my answer saying that there's hydrogen bonds between them because I just looked at the slides and it says that we have inverted complements to each other using so for example, if there's actually this is slide 28 of chapter 10, and it says there's inverted complement of each other of nucleotides, right? So I don't see how that would bond to itself unless there were hydrogen bonds involved.
I don't have the answer to that. I trust you. I don't have the answer to that. It could be hydrogen bonding. It could also be something about the phosphodiester bonds. I will actually look that up. That's a great question. Thank you. I'm going to look that up and get back to you.
Okay.
But just for the reasons, and I respect your answer of true, but I am personally going to go with false. Yeah. Any comments on that? I don't think the question is that specific. That's kind of what I was thinking too. I think it was kind of asking, do you know the difference between, or one of the big differences between RNA and DNA? You know, RNA can be double-stranded and DNA can be single-stranded. But just on the whole, that's a really important distinction. RNA is most often single-stranded and therefore doesn't make hydrogen bonds in most cases. I'm sure
Yeah,
it won't.
I'm just saying that the question could have been like worded a little bit differently to maybe like
Yeah.
clear that up. So I don't know, because I know like I was looking at the chat, a lot of people, I mean, like some people were confused. Like some people did put true and others false. So I don't know how this is gonna go down, honestly.
I will definitely ask Dr. Chiari about this, and I will look at that slide 25 as well as do some independent research. I think I remember reading something about, or maybe it was even in one of the guest lectures that we had about what actually does the like crimping and creation of the hairpin structures and like what holds it together. But yeah, thank you. That's a great question, and I will investigate that more and get back to you. Okay, so double helix molecule of DNA, what is the approximate ratio of A to T? Easy claps. 1. Exactly. 1 to 1. Exactly. I don't know, King. I'm sorry. Yeah, it must be a poll, or not pool of questions that everyone's Blackboard pulls from. We can go over your questions too if you like, and I can try to like restart this quiz and get the different questions. But yes, Kara and Casey had it right that the ratio is 1 to 1, or that the ratio is 1. Why is that? So the reason is because, excuse me, as we've talked about before, we have our double strands and our guys are connected to guys that are complementary. So wherever you find an A, it's going to be bound to a T. Wherever you find a G, it'll be with a C with a G. And so if you add all of the A's up, it'll equal the amount of T's. And therefore the ratio of A to T, even if it were 81 T's, it'll be connected to 81 G's. Therefore, divide 81 by 81, you get 1. That's not too hard. In some cases, or I remember Shargoff's data specifically, that it wasn't always 1 to 1. And that could be evidence that actually no, that's GC versus AT content. So that's a different thing, but you know, maybe he was just working with some funky data. Mutation. Yeah. And if the ratio is not 1, yes, actually, Kinga, that is a very good point. Thank you for reminding me. We can have mutations. This was originally ATTC, which obviously connected with G. But let's say that this guy mutated. What is the most likely nucleotide that it would mutate into? Don't be shy. Alex and Kate, so we have some C's. Alec has it. It would be an adenine. This is because guanine and adenine, even though they aren't complementary to each other, they are both purines. And purines are nucleotide bases that are a single hexagon connected to a second. And I'm not the best artist. Did I draw that with 6 sides? 1, 2, 3, 4, 5, 6. Yeah, we're good. So purines have their, I don't know the word for it. I'd say like doubly hexagonal, I guess, for lack of the proper term. And then we have our thiamine, which I'm never sure if I'm spelling right, and cytosine, which are pyrimidines. And pyrimidines are just the single hexagon. And the way I remember it, it's kind of convoluted. We have some answers in the chat. So most likely a base. Yes, Casey, exactly. Oh yes, the question was which, if this G here were to mutate, which of the four bases would it be most likely to mutate into? Obviously, like I'm not sure if it could mutate from G into G, so we can probably cancel that out. And the answer is A, because guanine and adenine are both purines. And this is a special kind of, or not special, but it is a mutation called, starts with a T. I'm going to let you guys guess it. That's another question in the pool. Oh, cool. Man, I am so just on top of everything. I'm too good. But yes, the way I remember the distinction between the shape of pyrimidines and purines is that, like I said, it's kind of convoluted, but pyrimidine is a long word and you'd expect a long word to be complicated, but no, the shape is very simple. It's just the single hexagon, unlike its complicated name. That's a really dumb way to remember it, but that's how I do it. But yeah, it is most likely that any of the four nucleotides are going to, if they undergo a mutation, it is most likely that they will stay within the kind of molecule that they are. A guanine will most likely mutate into an adenine because it retains this shape of having two hexagons. Because if it mutated into if it mutated into a thymine, for example, it would previously have this shape. And then all of a sudden, it would be stuck in here with only just that single hexagon. And that would cause a problem because then you'd have two single hexagons and these just don't bond together with hydrogen bonding well. Sorry, I just got, okay. Yeah, so it is most likely that a purine like adenine will mutate into a purine like guanine and vice versa. A pyrimidine will do the same, you know? So now that we know that, oh yeah, we already answered this question. Easy claps. It's 1. Yeah, there's a question that asks what purines are. This was easy. Okay. Boom. How did we do? We got 0 out of 10 points. Oh, because it's late. Okay. So yeah, apparently the answer was false, Joseph, but I really like your question, so I'm going to investigate it some more. But I do think it was just asking about the single-stranded nature of RNA. Would we expect it? Wait, would we be expected to only put 1? Yeah, because you know a ratio is kind of like, it's just a fraction, and well, I don't know. I don't, rounding questions. Blackboard is just stupid. So like, please don't ask me rounding questions. Try your best, and if you think you got it right, obviously you'll be able to look back at it and tell us if you got it right, and we'll change it for you. Yes, what's up, Joseph?
Yes, I was like looking it up while you're doing that last question, but I did find it. Like, there's like a Wikipedia page about the stem loop and stuff, and it does say it's hydrogen bonding.
Okay.
So
Okay.
that kind of makes this even more complicated because I guess it can go either way now. Because if hydrogen bonds are involved, I guess you could say they are connecting nucleotides. I guess you could say adjacent. I guess a better way to say the question would be say adjacent nucleotides. Because if it was a stem loop, then they wouldn't be adjacent nucleotides. It'd be like opposing nucleotides.
Yeah.
You know, but I don't know. Maybe we should talk to her. Should I email her about that? Like,
yeah, you know, like that's an excellent point, and I think you should email about it. Yeah.
okay.
Okay, that was one of the quizzes. Let's try and get some. Oh, can we do this question from quiz 9 I had? In maximum 3 words, explain why secondary structures such as loops and stems may occur in the RNA. Sam, do you want to take this one?
Yes, actually, I think somebody emailed us because they weren't sure if their answer was saved, and I thought that their response was really good. Let me find the email. Gosh, we have gotten so many emails the past few days that okay. So this person says inverted complement sequence. So basically, wait, what was the in maximum 3 words explain why secondary structures such as loops and stems may occur in RNA. So we know that RNA is all single, it's single stranded, like that's what makes RNA. And in order for RNA or DNA, you can have single stranded DNA, but the point is that when you have these strands made of your A, T's, G's, and C's, the only way that it can form a double strand is if it has a complement to it. And so if you have RNA that forms a loop or a stem, that means it has to fold over on itself. But in order for that folding to happen, for there to be binding between the parts that are folding, you have to have complementary sequences. Does that answer your question, Kinga? Yes, Kinga, that would be right. But that's more than 3 words, so I mean, like, I don't know if how she's awarding credit for that, but I would say like it seems pretty right to me. It's just more than 3 words. Oh, Vivian, do you want me to say like the answer, the 3 words that are the answer? Oh yeah, so we think that the answer would be inverted complement sequence. So I can type it in the chat. Repeated motifs. I am not sure. I don't think that'd be wrong. It sounds right. If she marks it as wrong, then you can always email us and we'll email all of us and we'll take a look at it. Okay, before we get lost, Casey asked if we could go over slide 42 from chapter 7. She asked like kind of a while back. So we'll go over that and then we will answer the more recent questions. But I just don't want Casey's request to like get lost in the chat. Garrett, do you want to go over that or do you want me to go over that?
You got it.
Okay, can you make it so that I can share my screen? Okay, cool. Let me see. Casey, is this the slide that you were referring to?
Yeah, that's right. But it is also 2 slides previous. So slide 40 is where I'm
Okay.
at.
All right, so did you want us to just kind of like go over it again, or was there something specific that you wanted to talk about from the cucumbers?
Well, I was reviewing this today studying. Anyway, I got sort of tripped up where it, on the next slide,
Mhm.
it has you take 1 and subtract it by 0.16 and also why we multiplied 0.08 times 2.
So the math right here is what's like tripping you up.
Yes.
Okay. So basically, so we are told that the recombination frequency is 16, right? And so because the recombination frequency is 16, that means that the frequency at which we are crossing over between our T's and our D's is 0.16. So that 0.16 accounts for T and D. which is why we have to divide it by 2 to get 0.08 for the frequency of each individual gamete. Does that part make sense so far?
Yes, but you're saying the gametes are looked at as A and T and then C and G?
Wait, sorry, can you say that again?
So if 0.16 is the recombination frequency, we're dividing it by 2, and you said we're doing that because we have to account for, you just said it, but I forgot, but I think you said like A and T and C and G.
So we have to divide by 2 because this frequency is it's the frequency for both RTs and RDs added together
Oh, the alleles up top.
Mhm.
Okay.
for the alleles. And so then you down here where they have where they get 0.42, it's kind of the same thing as up here, but we're just trying to find the non-recombinant gametes. So we just subtract 0.16. So that 1 minus 0.16 gives us the total frequency of our non-recombinants. So that would be the total frequency of non-recombinants for T plus D. And so because it's for T and D, we have to divide by 2 to get the non-recombinant frequency of just our T's or just our D's. Yeah, Garrett?
Yes, thank you. And so we divide by 2. Sam mentioned it, but we divide by 2 because we have a diploid organism here. Or damn it, I just gave it away. But I was gonna ask, what if we had like an octoploid individual? How much would we divide by? Yeah. Octo means 8 in Latin, I think. Anyway, yeah, sorry. I just wanted to mention that because someone asked me this. We learned this like a month ago, but even back then, someone asked me a really great question about that. So I just wanted to, mention it. Sorry to interrupt.
All right, so thank you so much, Garrett, for mentioning that. That was really good to mention. So Casey, does that part make sense now? Casey?
Sorry, I had an incoming call.
Sorry, no, it's all right.
So I was like trying to figure out how to get around that. It's like relatively clear. Maybe it's clear for everybody else. I just need to look at it a couple more times.
Okay, we can always go
Yeah.
over it again later on too if you would like for us to. Yeah, Alex, you raised your hand.
Yeah, so if there were like 3 letters on here, like 3 alleles, would that make it like would that change the ploidy and then we would divide by like 3 instead of 2? Or like, because if the ploidy was different, how would that change it? Like I know Garrett said if it was octoploid, but like what would that look like? Like if you had like 3, if the ploidy was like 3 instead of 2.
So if the ploidy was like 3 instead of 2, you'd have big T, little t, and then another T.
Oh,
So you
okay.
can have like a big T or a little t.
Thank you.
So Casey, do you want me to talk about slide 42 also? Or are you good on the cucumbers now?
Yeah, we can move on. Thank you.
Okay, no problem.
I just want to mention that cucumbers are members of the Cucurbitaceae and that they're a type fruit that is called the pepo, which is the same kind of fruit as a pumpkin and a squash and a zucchini. Yes, Cucurbitaceae.
That's cool.
I think Cucurbitaceae are rosids. Yeah, I think they're basil rosids. Anyway,
Oh, sorry. I was just gonna say Narj asked a question earlier. Oh, there was a question about the difference between the sugar of RNA and DNA, and I said the OH for RNA and H for DNA. Yeah, that would be correct because RNA has a ribose sugar and then DNA has deoxyribose, which means it's missing that OH from the ribose. I hope that answered your question. If it didn't, then just let me know. All right, I think we're caught up on all the questions in the chat now.
hooray. Do we have any more? I mean, not hooray, bad. Give us more questions.
I just wanted to clarify on something because on chapter 10, like she mentioned that there was like 3 different experiments that proved that DNA was genetic material. Do we have to know those?
Oh, yeah. Yeah. I think.
But I remember
Mhm.
her saying that she wasn't specifically going to ask questions about that on the exam, but now I'm like, I don't know if she will.
What do you think, Sam?
I was
can
gonna say,
remember.
I honestly cannot remember whether or not Dr. Chiari said you guys have to know it for the exam. But I mean, it doesn't hurt to know about them. I mean, I didn't take genetics with Dr. Chiari, but the professor that I took it with, she really liked to ask about the experiments because they were like pretty easy questions. And also, I think Garrett and I, we reviewed the experiments pretty extensively during our reviews. And sometimes Dr. Chiari likes to pull questions and concepts from our reviews. So there is a chance that she could ask about the experiments. I don't want to say that like she won't at all.
And like the 3 genetic experiments that we did learn about are like well, they're not the most clear cut, but they're the most like easy to remember. Because I don't even think, yeah, we didn't even go over like Meselson and Stahl and all the others. My professor also really liked the experiments. And oh, Gabby, yes, genetic mapping is going to be a huge part of this exam, like 2 and 3 point crosses. If that's what you mean. Is that what you meant, Gabby? Okay, yes. 2 and 3 point crosses. On my professor's exam, 3 point crosses were like a huge thing. And that's why I like care so much about it. But maybe Dr. Chiari won't. Anyway, they're important. You should know. And we have practiced them quite extensively. If she skipped the slide, then I don't see how it will be on the exam. Yeah, if Dr. Chiari didn't spend a lot of time on it in class, then maybe count on it not being so heavily present on the exam. But I think the experiments are cool. And if you want to talk about them, we can. If you could go over question number 1 from the weekly review 10, not the same question, but similar. Question 1, that was the 2 point cross, right, Usman? Okay, yes, I can look for another one. Difference between 2 and 3 point crosses. So 2 point crosses are really easy because you don't have to worry about
the middle gene.
Yeah, the middle gene and double crossovers. But yeah, with 2 point cross, I guess we have a request for 2 point cross. So I will look for one. There's an ant on my desk and I don't want to squish them. So I blow them away. Yeah, if you guys can think of some questions while I look for another 2-point cross, that would be. I think I have one. Where is this? Actually, no, let me look for a better one. Ooh, could this be it?
Garrett, do you just want to do the one that Kinga talked about, 28 from the book?
Yes, I am on a device that does not, actually, no, I put that in the drive, so it should be accessible.
I can also copy and paste the question. Kinga, is it from chapter 8? Or sorry, chapter 7? Okay. Because Casey also suggested slide 49 from chapter 7, but I figure we've seen that one already. So it doesn't hurt to see another. Garrett, if I send this as a picture in the chat, will you be able to see it?
Yeah, sure. Maybe, I don't know, hopefully.
I guess we're gonna find out.
Okay. Yes, we will find out. Oh yeah, I can download it. I forgot you can send files on Zoom.
Gosh, I always forget to.
Sam, you sound like a robot. Oh my God, so many things are opening up. What's happening? Ah. Okay, let's see. No, open, please. Okay, this is a long question, but we can do it. So a series of 2-point crosses were carried out among 7 loci, producing the following comment. What's happening? What?
Oh no.
Where'd these? I love these birds, but where'd they come from? Oh no, what's happened? Excuse me. Okay. Hopefully it won't. Hopefully the birds won't take over again. So a series of 2-point crosses. Okay. Producing the following recombination frequency. So we have A through G. Map the 7 loci, show their linkage groups and the order of the loci in each linkage group. So what's the first thing do you think we should look for? Yeah, the ones with 50. So we see a 50 here. 1, 2, 3, 4, 5, 6. Wow, that's a lot of 50s. Okay, so what is a 50%? Oh my, there are even more. Goodness. What does 50% recombination mean? What does that tell us about the distance between B and D, B and F, B and G? That they're unlocked, exactly. That they're unlinked. And what does it mean to be unlinked? What does that mean for their distance? The amount of space between them. Yes, it could be that higher percents of recombination correspond to higher or larger distances between, yes, longer distances between the 2 genes. And that distance could be so far that like they could be just like super far away from each other, or this distance could be so far that they're actually not even on the same chromosome. And so, yes, 50% recombination displays unlinked status between two genes, C and D, C and F. And so we see this a lot with our C. We see that C is very commonly 50% further or away. And we see it a few times with D. Actually, we've seen it with C and F. We've seen it with D and F. We've seen it with E and F, F and G, B and F, and A and F. So like everything is unlinked with F. So you can consider F kind of in its own world. And this is how I do it. I just like try and isolate. Oh, I accidentally erased it. I try and isolate the different alleles or genes and then build my map from there. Up to 50%. Yes, 50% is the maximum frequency of recombination. And I used to know how to explain it, but I no longer do. So I won't try to contrive it and waste your time. But okay, so, and we see that A and B, I'm gonna stop erasing these, A and B, A and E. So A is also very commonly far away from other things. B is also commonly far away from other things. So we can see that A and, or F is in its own world. A is in its own world relative, actually, I wish this weren't so cluttered of a screen. Sorry. Switch colors. So F is in its own world. A is unlinked from B. So A is over here, and that is actually an A. And B is over here. A is also disconnected from C. A is disconnected from E. So I'm just like looking at all this stuff. And then A is also disconnected from F. But A is kind of close to G, or it's really close to G, and it's kind of close to D. So it's really close to G, and kind of close to D. And once we have this like preliminary kind of, excuse me, orientation, then we can look at the different letters and further elaborate on our fungi. Yes, Joseph. So yeah, we know from this that F is very far away. So we could even qualify it sooner and say that F is over here. It's far away. And we know that B is also far from D, so that's good. We know that since A is close to D, A is far from B, D is also probably far from B. So on one, we will have AGD and the other CBE. We don't know yet, but that's a good guess so far. We know that B is far from D, B is far from G, that's good. We know that A is close to these, and these are far from A, so they're also probably far from the other two. And we can move on, C and D. So C is far from D, that's good. C is kind of close to E. And we see here that C, I'm gonna stop being so inconsistent with my colors, I'm so sorry. So now we're looking at C, and we know that C is kind of close to E, not really, but it's not 50. And we know that C is close to B. So we know that, and we can also look at this. So we're kind of just like creating a preliminary map and then using further numbers to like qualify it and try and make it more and more accurate. So we know that C is close to B, and C is kind of close to E. So let's try and look at what's near E so we can see how far away it's going to be. So we know that C is kind of close to E, but how is E relative to B? Because these are kind of in the same group. So B and E, not terribly far. We know that E is closer to B because it has a smaller number than it is to C. So actually, we have just found out that B is closer to E than it is to C. and C is also very close to B. And I think that's all the letters. Actually, no, we can further elaborate on this section. So I think this section is good for now. And we know the distance between C and B is 10. The distance between B and E is 18. And here we can see that this distance between C and E doesn't add up to the interstitial distances that we have from C to B and then B to E, because this distance between C and E is 26, which is not equal to 10 plus 18. Excuse me. And so now that we have sorted out this section and we know the relative distances, let's try and sort out this section. So we have A, G, and D. So A is, I'm going to switch colors again. So A is really close to G. A is kind of close to D. So how is D relative to G? D relative to G is 8. So D is closer to G than it is to A. So this is probably good because G is really close to A. G is really close to D. And D is closer to G than it is to A. So this is also probably good order. So we have that the distance between A and G is 4. The distance between G and D is 8. And the distance between A and D, actually this time it does add up to 12. The distance between D and C is too great for us to determine. And so we have our map. I'm going to draw it down here. We have our overall map. And actually, someone probably just said what I was going to say. Yes, awesome. Go King, go. King is the best. Okay, but yes, we have this linkage group here, and we know the distances between the genes on this or in this group, and we have the linkage group here and a linkage group here. F is all by its lonesome. So to answer the question, map the 7 loci, show their linkage groups 1, 2, and 3. The order of the loci in each group, we did that. And the distances between the loci of each group. So we know that the distances between this group and this group is 50. This group and this group is 50. And this group and this group is also technically 50. But this 50 doesn't indicate anything about, like this F could actually be over here, but still 50. And they're just too far away. We don't really know. F happens to be isolated from A and B and C, excuse me, and D and G. So it's just too far gone. We don't know whether or not it's over here, whether or not it's over here, whether it's on different chromosome over here, you know. But within the linkage groups, there's a lot of color on the screen, but we did it. All right. Is AGD on one chromosome and CBE on another? Or Usman, yeah, I think I just answered that. They could be on the same chromosome and just like at the other, like at opposite ends, or they could be on an entirely different chromosome. We can't really tell because we don't have enough information. No, that's the only one I get. Okay. F could be on another chromosome, right? Yep. Or their linkage groups on different chromosomes. All the linkage groups could be on different chromosomes. They could be on the same one, just really far apart. So it's all on one chromosome. They could be on the same chromosome or they could be on different ones. We just don't have the information to tell. All right. How do we determine? I don't know the answer to that. Do you know, Sam? I think we need
Typically
more
it's
info.
assumed that if it's 50 or more, it's different chromosomes.
Okay.
Or that's the way that I learned it.
All right. Do we feel like doing a 3-point cross? What's the time actually?
It is 8:28.
Okay. How long do we want this to be?
I was just gonna say we could go until like 9 if people still have questions.
Yeah, I'm good with that. Okay, so good for you, Usman. Good for you. So what are the major aspects we should focus on? Chapter 11. What is chapter 11? Let's find out. This is not the right class. This is the right class. And I wrote it in the announcements and I color coded it. So chapter 11 is yellow. The differences between varying genomes, so like mitochondrial, chloroplastic, and nuclear DNA. We can pull up the slides on that if you'd like. Yeah, chromosome structure. Maybe? Or is the chromosome structure a different chapter? Do you want us to pull up the slides on that? Okay, no problem. Yeah, this is some notes for exam 3 if you haven't seen this announcement already. So apparently this purple business might not be so prevalent on the exam. So take that with some apprehension. But I thought it was going to be important. It might not be though. So King asks, mostly 2 and 3 point cross stuff, shuffling will be heavily tested. That's like Sam and I have said, we had different genetics instructors, and with these reviews and stuff, we're just like trying to talk about the stuff that we think is the most essential to doing well. And that's kind of like difficult because we had a different instructor and we aren't exactly sure what Dr. Chiari is going to test on. But these are the most important concepts. And we definitely could do another 3 point cross. And it's been asked for 3 times actually.
Oh, sorry.
Oh yeah, what's up?
And I was just say while you're pulling up another 3 point cross question, one thing that we do want to let you guys know is like we literally have no idea what the exam looks like. Like we don't get to see it before you guys do. In fact, we don't see it until you guys see it or even until after you guys take the exam. So we're not sure which topics are going to be really heavily tested on or what kinds of questions Dr. Chiari is going to ask. And so the things that we go over during the reviews are things that, like Garrett said, we think are important or things that we think can be a little bit more confusing or things that we felt weren't discussed enough during class. So like our reviews by no means are like all inclusive of everything that you should know.
Okay, I found a jank one from the internet. So let's see if it's worth our time. You know what? I believe in us. Let's try it. Okay, so what are we looking for, firstly, when we're doing 3 point crosses? The middle gene, non-recombinance, just like in general. Yes, we do need to find the middle gene and the non-recombinant progeny. Yes, exactly, Casey. We're looking for the order of the genes and the distances between those genes. And so the first step to doing that, like we said, is finding the middle gene. And just like Kinga said, the way we do that is by comparing the non-recombinant individuals to the individuals who exhibited double crossing over. Oh wait, can we even do, so recombinant for LSI, V and CV, V and CT, CV and C. Can I even interpret that? Oh yeah, here their genotypes right here. Oh wait, this is kind of confusing. Oh, so this is actually even extra information. So we can pretty much just look at this. This is kind of cheat code, cheating mode, doing the work for us. Yeah. Yes. Caroline says double crossovers are the last two. So the way you can tell, so what we're going to do is compare some parental individuals with some individuals who exhibited double crossing over. And the parental individuals are the most numerous, obviously. There's some big numbers. And then the double crossing overs are the least numerous progeny. And I should change color. And the reason we know this is because of how likely it is for crossing over to occur. It's very likely that crossing over doesn't occur, and that the organisms they display the parental genotype, and it's very unlikely that they will cross over twice within the same chromatid. So, and then we can, you guys are already so far ahead of me, we can compare these. So we have these genotypes and we can compare just one of them to another. So let's compare these two. So we have V, CV plus, CT plus. And then we have V, CV plus, CT. So what's different about these two? V and V are the same, CV, CV are the same. It's that's the same, and therefore it is the CT gene with the possible plus allele. That is the central gene. Do we want me to talk about why we know that's the central gene? Like go over the rationale? You remember the traveling buddies? Okay, good. We remember the traveling buddies if you've watched any of the reviews or videos since then. So we know that the CT gene is in the middle. I like to rewrite these. It is more palatable for me personally. So the genotypes. Ooh, I'm going to do that. What is happening? Oh, very cheeky, very cheeky. But I never do it in the right spot. Ugh. Very clever, Garrett. So we have, oh, damn, I don't even remember what they were. Okay, let's restart. Yeah, I was thinking about that. Okay, never mind, forget it. So we have our guys, and actually I am going to do that to make more space for myself, but it shouldn't be black, it should be white. Sorry, technical difficulty. So anyway, as we were saying, CT is in the middle. So we have the V gene, the CV gene, and the CT gene, and we know that CT is in the middle. So this is our map so far, and I'm going to rewrite it. Sorry for constantly annoying you with my shenanigans. Ah, goodness gracious. We are patient. We are patient. So draw and make the color not white. So V, CT, and CV. We know this is the order, and it could be that CV is actually over here and V is actually over here, but we don't have enough information to determine. Like, for example, if we had another gene, G, and we knew that G was somehow closer to CV, then we'd know that, and we knew that G is over here, and we know that CV is over here and V is over here, but we don't know that. So anyway, can we do it without rewriting? Yes, I think Sam said she does it without rewriting. Yes, I prefer not to rewrite. Okay. Okay, no problem. So we have our guys. And so we have the relative positions, the order, but we need to learn the distances between the genes. And so we can write this. I like to write this as having a cross between the parental individuals. So we know that these are the parental genotypes. And we have V with CT, or I guess you don't want me to rewrite it. So V with CV plus and CT plus. It can be easily, yeah, depends on your preference. So we have, this is one parent, the 580 individuals, and then the 592 individuals are V plus. Why wouldn't you rewrite it now that I'm doing it now? Anyway, actually, I also messed it up. But it's okay. We are patient. We have V plus CV CT. I'm going to rewrite it because I don't think I could do it without rewriting it. Yeah. Sorry if you wanted me to rewrite it, but I just don't understand it without rewriting it. Because this is how I do it. I put them in the center, put the correct one in the center anyway. And then I can show the individual crossing over events. So this is going to be our single crossover 2. And this one, our single crossover 1. So we need to find the distance between V and CT by finding the recombination frequency at the first locus. Excuse me. And then the distance between CT and CV with the second locus. So first locus. The first locus we'll see crossing over right here. And so we can write the cross. So we can write out the genotypes that we saw that had the crossing over at this locus. So we'd have V, we can follow the cross with CT, CV. And then we can also have V plus with C plus T plus, excuse me, or C plus, CT plus CV plus. So V plus. And what do we notice about this cross? Is the process similar for 2-point crosses? I mean, we just did a 2-point cross. Do you think it's similar? I don't know. I don't think so. Yes, exactly. The parents are in trans configuration, also known as repulsion. Because usually when we see parental genotypes, they're all nice and pretty, and they have, no plus, no plus, no plus, all plus, all plus, all plus. But these parents have some different alleles. The yellow ones are plus alleles, and the red one is the just regular allele. So these parents are in trans. And we see that the result of this cross creates progeny that are in cis configuration rather than the parental being in cis configuration. Anyway, so we have this, we have the genotypes, and now we can look at where these individuals are and find the number that they occur in. So we have just the plane is right here, and that's 89. You know, no plus, no plus, no plus, no plus, no plus, no plus. So we have 89, and then all pluses is right here with 94. We can combine them. 89 plus 94. What's the next step? The 3 and 5, exactly. When we are looking at the amount of individuals that crossed over at this first locus, we have to consider double crossovers as well, because they double crossed over. They crossed over at this locus and this locus. And because they did cross over at this locus, they are considered when looking at single crossover 1. Double crossovers always seem to be 5 plus 3. Like, you think the people who can make these questions are more creative than that. So, and then we divide this by the total number of individuals, which is 1, 4, 4, 8. We see it right here. And this is a new example to me. So if someone could do the math on that, that would be very helpful. So I'll send it on previous example sheet. This would be CTV connection. Oh, thank you for the math. CTV connection. Yeah, exactly. It's crossing over at this locus, which is in between the V allele and the CT allele, or gene, excuse me. 0.132. Thank you very much. 0.132. And yeah, if you don't like to rewrite your genotypes, Sam has an excellent guide at the bottom of the week 10 review that walks you perfectly through how to do a 3-point cross. And so we have found the distance between the V and CT allele. We know that it is 0.132. Now we find single crossover 2. Yes, it depends on the gametes you're given. Because if we are given this in any different combination, like if this were V plus and then regular CV, this whole cross would be different. Because the middle gene might not be the same. So yeah, it all depends on what you're given. You just have to try your best. So I'm gonna also get rid of these. Okay. So we had that crossing over. And now let's consider the second locus. So we can follow our cross. We had this, which is V CT plus CV. We also had VT plus CT CV plus. Can you leave it up? Yes, absolutely. Sorry, I should have done that on the quiz too. I will look at the recording really fast once it's done and get you that so you can email Dr. Gary. So we have these genotypes. Also, I'm going to erase this. So we have these genotypes and we can look in our progeny for their number. So so we have V, CT plus CV, which is this guy, and that's 45. And then we have V plus CTCV, V plus CT, where you at? Oh, it's also C2 plus. Wait, no, what? Did I write that right? It's probably this one. Oh yeah, I just had it in the wrong order. That's why it's helpful to rewrite. I guess, or I guess if you didn't rewrite, it would also be compatible. Anyway, so we have these guys and we can add them together now. 45 plus 40 divided by 1448. And that gives us, what does that give us? Don't disappoint me. Hooray, thank you. We also add the 5 plus 3. What does that give us? 0.064. So we have this distance, the recombination frequency between the CT and CV gene, so 0.064. And we have just completed another 3-point cross. This is our overall answer. And that was easy claps. It was also in trans. Thank you, Mayad, for coming. Mayad's always here. What a champion. I appreciate you. But yeah, that was in trans and we hadn't even talked about that in class, but we still did it. No problem, because we're the best. I didn't expect it to be in trans. Would anything be different if it was cis to trans? Yeah, so if your parents are in cis configuration, then you'd have these 580 individuals would be like the CV CT would be the 580. And then V plus CV plus CT plus would be the 592. And trans configuration, trans is a Latin prefix that means like across. And that refers to the fact that the alleles for just these individuals would actually be V, CV plus, CT plus, and then V, CV, CT. And it's in trans because across this space from one chromatid to another, or one homologous chromosome to another, across that, they aren't the same. That's what the trans refers to. Some questions in the chat. Equation would stay the same. Yes, exactly. You'd just be dealing with like different actual combinations of letters. So as long as you follow this process, it's all good. And so that's trans, but cis would be, I don't actually know what cis means, but just in relation to trans, it means the same. Cis prefix meaning. Let's find out. Yeah, on the same side as is what it literally means. Cis means same side. And so in that instance, we'd have V, CV, CT with V, CV, CT. And across this, I guess they're like on the same side because they're pretty much the same. And the parents would look like this instead of like this. Oh, thank you, Kinga. Do you speak Latin? Yeah, so for chemistry, cis is, yeah, that's where I was originally exposed to cis versus trans is in organic chemistry. Yeah. Ew. Relatable. Okay, so another killer 3-point cross. Very good job. I didn't hate organic chemistry, but I have to take it again. So I'm bitter about it. Do we have any more questions? We have 7 minutes. Take it with Dr. Ward. Thank you, Vivian. I will try to take your advice. I have to take it next semester. Actually, I'm going to register tomorrow. Thank you guys, saving my life. Probably get a terrible professor otherwise. I do mind driving to Manassas, but thank you. What's the coolest classes, or which class are you guys excited for taking next semester? If you are taking classes next semester. Physical chemistry, anatomy. Ooh, I'm also taking anatomy, Caroline. Oh my God. Or Kara, excuse me. Again, where does the goat? Anatomy. Oh, we're all taking anatomy together. Wow. Oh, research semester. How did that go with the application? Animal physiology. Is that like bio 3? I don't remember the number. Hunnell, thank you. I'm gonna write these down. So Dr. Ward for OChem and Hunnell for anatomy. You haven't heard back yet either, okay. 326, okay. Yeah, I was just looking at it because I'm planning out my bio electives for future semesters. Animal physiology. Oh, I can cut the recording or pause it listed. Okay, so we are back. And King of Midnote. So someone contacted Dr. Pieravon, the oncology lecturer, asked if she would be offering a cancer class for undergrads, and she said she's working on it and will let us know by the end of May. And if we're interested, we can post it on Discord. All right, that's awesome. I was considering that. I think it was like 492 or something, she said. I don't remember. But yeah, oncology is cool. 592, okay. Yeah, she's working on unveiling it for undergraduate students because it's currently a grad only class. So it is 8:59. And so I think we are going to maybe, just maybe, cut the recording. Ooh, Ganari. I should look that up. Oh, well, thank you guys for coming. And yes, exam tomorrow morning, regular class time. We will be around if you guys need some help. No, thank you. Thank you.
Thank you guys so much for coming and for having questions. I'm always worried that people won't have questions and we'll just be like sitting here in awkward silence, but very glad that you guys came with many questions.
Oh yeah, and Joseph, if you get that answer, then let us know. Observation. But yeah, thank you all for coming. And if you have any questions before the exam, we're here. try your best. I sincerely believe in you. You guys are going to crush it. This is one of the easier exams, so I've heard. But yeah, thank you all for coming. Have a great night. Try to get some good sleep. Don't drink coffee or eat chocolate because it will increase your heart rate. I can tell you about actually, Sam could probably tell you more about the effects of coffee on your biochemistry. But anyway, sleep well and yeah, eat some cucumbers. All right, good night, everyone.
Bye, guys. Good luck.
Oh yeah, Vivian, I'll upload it tonight. Like really soon. But yeah, good night, everyone. Thank you.